Good evening, everyone. So today is the 13th and the last live session for this course. I share my screen and we can start today's session. Give me a minute. Yep. Can one of you unmute and let me know if I'm audible and if my screen is visible? I assume I'm audible.
Yes, ma'am, you're audible.
Okay. Thank you. So good evening, guys. Today is the 13th and the last live session of this course. So as you know, in this course, which is Genetic Engineering Theory and Application, taken by course instructor Professor Vishal Trivedi from IIT Guwahati. And in these live sessions, we basically discuss some of the previous year assignment questions. We not only discuss the questions, we try to discuss the concepts behind those questions. So it's easier for us to solve similar kinds of other questions. Okay. So we try to keep the sessions as interactive as possible. But in case if you're not able to ask now, you can always ask me later also. And all these slides will be uploaded and all the videos, video recordings will also be uploaded on YouTube. So moving to the first question for today, which of the following is the start codon? Anybody?
AUG, first option.
Yeah. So yes, the right answer is AUG. So we have talked about transcription and translation in one of the previous classes, and we know about central dogma as well. That is the flow of information in the cell that starts at DNA. It replicates to form more DNA, and then it gets transcribed to mRNA, messenger RNA, which ultimately gets converted to protein, which is called translation. Okay, and the proteins are basically the warriors in the cell. So what does they do most of the work in the cell. Okay, transcription is carried out by RNA polymerase, replication by DNA polymerase, and translation obviously by ribosomes. And there is a lot of other factors involved as well. We need tRNAs or transfer RNAs as well. So information does not really flow in the other direction. Okay, this is the molecular version of the incorrectness of inheritance of acquired characteristics, and changes in the protein do not affect the DNA in a systemic manner. Okay, if there are mutations in the DNA, in the genes, then that will affect the protein. But if there's something happening to the protein and the last step, some misfolding or anything, that will not affect the DNA, right? So it is not information does not flow in the other direction. The only time when information does flow in the other direction is via reverse transcription. Where RNA, so it is done by the enzyme reverse transcriptase, where RNA gets converted to DNA. So that is the only time when a part of in this central dogma part of it is in the opposite direction. So if we talk about reverse transcription, give me a minute. So if we talk about reverse transcription, so that is, as I said, a few exceptions of the central dogma. And most importantly, some RNA viruses, which are the retroviruses, they make DNA copy of themselves using the enzyme reverse transcriptase. And this DNA copy is what they incorporate into the host chromosome. And that becomes a permanent feature of the genome. So the DNA copy inserted into the genome is called a provirus, and this represents a flow of information from the RNA to DNA. Okay, and closely related to retroviruses are retrotransposons. Okay, so what are exactly transposons? Transposons are nothing but it's called basically jumping jumping genes. And what are retrotransposons? So its transposon is basically a DNA sequence that can move within a genome. So they can create or reverse mutations and alter the cell's genetic identity and genome size. Okay, so transposons move through a DNA intermediate as opposed to retrotransposons which move through an RNA intermediate, right? So if we talk about the differences between the transposons and retrotransposons, they are both basically they differ in the way how they move and the mechanism they use. So retrotransposons, they use a copy and paste mechanism to move, duplicating the element into a new genomic location via an RNA intermediate. So retrotransposons are one proposed evolutionary mechanism for gene duplication, whereas transposons, they use a cut and paste mechanism to move. So they can cause mutations, alter gene expression, induce chromosome rearrangements, and increase or decrease the amount of DNA in the genome. Okay. So there are some other differences between retrotransposons and transposons as well. HIV-1 and some other human retroviruses, they behave like retrotransposons. Okay. So they are basically sequences of DNA that make RNA copies of themselves, which then get reverse transcribed into DNA that inserts into new locations in the genome. So unlike retrotransposons, retroviruses, retrotransposons always remain within the cell. They lack the genes to make the protein coat that surrounds the viruses. So that is what is the only differing factor between retroviruses and retrotransposons. A little bit into prions, which is a very hot topic in today's research all over the world. A prion is an infectious protein, right? So you might have heard about infectious pathogen, infectious bacteria, infectious virus, but it's an infectious protein. So prions are the agents that can cause mad cow disease, which is bovine spongiform encephalopathy, chronic wasting disease in deer, elk, scrapie in sheep, and this syndrome in humans. I'm not exactly sure how to pronounce this. So these diseases cause neural degeneration, right? And why are prions so infective? Basically, so what happens? They can cause, as I said, fatal brain disease in humans and animals. So they are abnormal forms of the prion protein (PRP) that are present in all healthy humans and individuals, animals. Okay. So when these PRP molecules are converted into the abnormal form, they can cause a neurological disease. Okay. So the symptoms are approximately those of Alzheimer's syndrome, accelerated to go from onset to death in about only 1 year. Okay. And it is, disease is very hard to catch and very rare. That is a good thing. And they usually have a long incubation time. No cure is known till now, and not enough is known about it either to, so that we can do a good job of preventing it. Okay. So the prion protein is normally present in the body, as I said, and like all proteins, it is properly folded into specific conformation, a state called PRPC. Now these prion diseases are caused by the same protein folded abnormally, a state called PRPSC. Right? So this PRPSC can bind to a normal PRPC protein and convert it to PRPSC as well. So basically, as people say, one bad apple can ruin the entire basket. So even if one part of the protein is not folded properly, it can ruin the other normal PRPC proteins as well. And this conversion can spread throughout the body, causing the disease to occur. So it's also a form of inheritance that does not involve nucleic acids. Okay. And they can be apparently transmitted through invasive medical procedures, exposure to contaminated food, infected tissues, body fluids, etc. Okay. And that is how prions are spreading. So RNA, we know it plays central role in the life of the cell. We are mostly going to look at its role in protein synthesis, but RNA does many other things as well. They can store information like DNA, and they can also catalyze chemical reactions like proteins. So RNA can also act as a catalyst. So one theory for the origin of life has it that it started out with RNA only. DNA and proteins came much later. So this theory gives rise to the fact, the hypothesis of the RNA world. Okay. So what does the RNA world say? So the RNA world is a hypothetical stage in the early evolution of life on Earth, where RNA molecules were the primary means of storing genetic information and catalyzing chemical reactions. Okay. So this hypothesis suggested that RNA came before DNA and proteins in evolution. It posits that RNA molecules were able to replicate themselves without the help of other molecules. So Walter Gilbert coined this term RNA world in 1986, and there is strong evidence that RNA world existed before DNA and protein-based life. For example, the ribosome and the pre-mRNA splicing machinery can be explained by viewing them as descendants of RNA mediated interactions. Okay, however, there are still challenges to the RNA world hypothesis. It's not exactly clear how all the components of the RNA were available in a prebiotic pool without the prior existence of evolved macromolecules. Right? So RNA protein hybrid structures are also involved in protein synthesis, which we know as ribosomes, right? Splicing of mRNA, telomere maintenance, guiding ribosomes to the endoplasmic reticulum, and other various tasks. So recently it has been found that very small RNA molecules are involved in gene regulation. So if you talk about the different types of RNA, mRNA or messenger RNA is a copy of the gene that is being expressed. Okay, so groups of 3 bases in mRNA called the codons. They code for each individual amino acid in the protein made by the gene. So in eukaryotes, the initial RNA copy of the gene is called the primary transcript, which is modified to form mRNA. Then we have the ribosomal RNA. 4 different RNA molecules can make up part of the structure of ribosome. They perform the actual catalysis of adding an amino acid to a growing peptide chain. Then we have tRNA or transfer RNA, like small RNA molecules that act as adapters between the codons of messenger RNA and the amino acids they code for. Now, RNA contains the sugar ribose and DNA contains deoxyribose. RNA contains uracil, DNA contains thymine. RNA is usually single stranded, DNA is usually double stranded. RNA is short, one gene long at most. DNA is long, containing many genes. Okay. Now, if we talk about transcription, it's basically the process of making an RNA copy from a DNA, single gene, and genes are nothing but specific regions allotted in the DNA of a chromosome. So this entire mechanism is carried by the enzyme protein RNA polymerase, and there are several forms of RNA polymerase. In eukaryotes, mostly most genes are transcribed by RNA polymerase 2. So the raw materials will be the 4 ribonucleoside triphosphates ATP, CTP, GTP, UTP, adenosine triphosphate, cytosine triphosphate, guanine triphosphate, and urine triphosphate, and it's the same ATP as is used for energy in the cell, right? So you might be very familiar with the term ATP. Now, as with DNA replication, transcription proceeds 5' to 3'. So new bases are added to the free 3' OH group. And unlike replication, transcription does not need to build on a primer. Instead, transcription starts at a region of DNA called a promoter, and for protein coding genes, the promoter is located a few bases 5' or what we call upstream to the first base that is transcribed into RNA, right? So promoter sequences are very similar to each other but not identical. So if many promoters are compared, a consensus sequence can be derived. So all promoters would be similar to this consensus sequence but not exactly identical. So transcription starts with RNA polymerase binding to the promoter, and this binding only occurs under some conditions when the gene is on, like it needs to be transcribed. Various other proteins or transcription factors help the RNA polymerase bind to the promoter. Other DNA sequences further upstream from the promoter are also involved. Now, once it is bound to the promoter, RNA polymerase unwinds a small section of the DNA and uses it as a template to synthesize an exact RNA copy of the DNA strand. Now, the DNA strand used as a template is the coding strand. The other strand is the non-coding strand. So, the RNA is made from 5' end to 3' end. So, the coding strand is actually read from 3' to 5'. And RNA polymerase proceeds down the DNA, synthesizing the RNA copy. Now, in prokaryotes, each RNA ends at a specific terminator sequence, and in eukaryotes, the transcription doesn't have a definite endpoint. The RNA is given a definitive termination point during RNA processing. So it's a schematic of transcription. It's graphics. So this is the promoter, RNA polymerase, with the help of various factors. We'll not go into too much details. It starts, then we have the initiation process, then elongation, and then finally termination. Okay. So after transcription, in prokaryotes, the RNA copy of a gene is the messenger RNA ready to be translated into protein. Now, in fact, translation starts even before the transcription is finished in prokaryotes. Okay. In eukaryotes, the primary RNA transcript of a gene needs further processing before it can be translated. So this step is known as RNA processing. Also, it needs to be transported out of the nucleus into the cytoplasm. So in prokaryotes, we do not really have this concept of a proper boundary, uh, c- nucleus containing boundary. So that's why even when transcription is still going on, translation can start at the beginning. But in case of eukaryotes, uh, after transcription, there is a process, there is a part step called RNA processing, after which it is translation. So what are the steps in RNA processing? It adds a cap to the 5' end, adds a poly A tail to the 3' end, and it splices out the non-coding regions or the introns. Now, what is capping? RNA is inherently unstable. Okay, which is also one of the reasons why people believe that we have shifted from the RNA world to something to DNA, which is much more stable. Okay, so RNA is unstable, especially at the ends. So the ends are basically modified to protect it. So at the 5' end, a slightly modified guanine 6,7 methyl G is attached backwards by a 5' to 5' linkage to the triphosphates of the first transcript base. And at the 3' end, the primary transcript RNA is cut at a specific site and 100 to 200 adenine nucleotides are attached, which is known as the poly A tail. So these A's are not coded in the DNA of the gene. These are added after the transcription process, before translation. Now we have talked about splicing out introns. What are introns? Introns are regions within a gene that don't code for protein and don't appear in the final mRNA molecule. So protein coding sections of a gene called exons are interrupted by the introns. So the function of introns remain unclear. They may help in RNA transport or in control of gene expression in some cases, and they may make it easier for sections of genes to be shuffled in evolution. But no generally accepted reason for the existence of introns still exists. Okay, so there are a few prokaryotic examples, but most introns are found in eukaryotes. Okay, and some genes have many long introns. The dystrophin gene, the dystrophin gene is quite popular because the mutation in the dystrophin gene can cause muscular dystrophy. It has more than 70 introns that make up more than 99% of the gene sequence. However, not all the eukaryotic genes have introns. Histone genes, for example, they do not really have introns. How does the intron splicing happen? It's a complicated process. So they are removed from the primary RNA transcript while it is still in the nucleus. So they are basically spliced out by RNA protein hybrids called spliceosomes. So the intron sequences are removed and the remaining ends are reattached so that the final RNA consists of exons only. So we have, if it's talk about summary of RNA processing, RNA polymerase produces a primary transcript and RNA exact RNA copy of the gene. A cap is put at the 5' end. RNA is terminated and poly A tail is added to the 3' end. All introns are spliced out, and at this point the RNA can be called messenger RNA. It is then transported out of the nucleus into cytoplasm where it is translated. Now, proteins are composed of one or more polypeptides plus some additional small molecules called cofactors, coenzymes, prosthetic things as well. Now, uh, polypeptides are linear chains of amino acids, and after synthesis, the new polypeptide folds spontaneously upon itself into its active configuration and combines with the other necessary subunits to form an active protein, right? So all the information necessary to produce the protein is contained in the DNA base sequence that codes for the polypeptides. Now the sequence of amino acids in the polypeptide is known as its primary structure. Now there are 20 different amino acids coded in DNA. They all have an amino group, NH2 group on one end, and an acid group or COH group on the other end. So attached to the central carbon is an R group, which differs for each of the different amino acids. So when polypeptides are synthesized, the acid group of one amino acid is attached to the amino group of the next amino acid, forming a peptide bond. You can see over here, CONH. So these are different amino acids, the different features, properties and all. Some are hydrophilic, some are hydrophobic, some are nucleophilic, some are acidic, aromatic, basic, etc. Moving on to translation, it's basically the conversion of mRNA into protein, which is done by ribosome and RNA protein hybrid. So ribosomes are composed of two subunits, large and small, and ribosomes bind to the translation initiation sequence on the mRNA, then move down to the RNA in the 5' to 3' direction, creating a new polypeptide. The first amino acid in the polypeptide has a free amino group, so it is called the N-terminal, and the last amino acid in the polypeptide has a C-terminal free acid group, it is called the C-terminal. Now, each group of three nucleotides in the mRNA is a codon which codes for one amino acid. So transfer RNA is the adapter between the codon, three bases of the codon and the corresponding amino acid. So if you see, this is the structure of a transfer RNA. They are short RNAs that fold into a characteristic clover leaf form pattern, and some of the nucleotides are modified to become things like pseudouridine and ribothiamidine. So each tRNA has three bases that make up the anticodon. So these bases pair with the three bases of the codon on mRNA during translation, and each tRNA has its corresponding amino acid attached to the 3' end. So set of enzymes, the amino acid tRNA synthetases, are used to charge the tRNA with the proper amino acid. So some tRNAs can pair with more than one codon. The third base of the anticodon is called the Wobble position, and it can form base pairs with several different nucleotides. So the first two positions are mostly constant. The third base will be the Wobble position that will change the game. Now, how does translation occur in prokaryotes? Ribosomes bind to specific translation initiation sites, and these can be several different sites on a messenger RNA. A prokaryotic mRNA can code for several different proteins. So translation begins at an EUG codon or sometimes a GUG, and the modified amino acid N-formyl methionine is always the first amino acid of the new polypeptide. Okay. In eukaryotes, ribosomes bind to the 5' cap, then move down the mRNA until they reach the first AUG or the start codon. Okay, and it also codes for methionine. The translation starts from this point. Eukaryotic mRNAs code for only a single gene, although there are few exceptions, mainly among the eukaryotic viruses. Now, if you have to know, the translation does not start at the first base of mRNA. There is an untranslated region at the beginning of the mRNA, the 5' UTR untranslated region. So the initiation process involves first joining the mRNA, the initiated methionine tRNA, and the small ribosomal subunit. And several initiation factors, additional proteins are also involved, and the large ribosomal subunit then joins the complex. Then the ribosome has two sites for tRNAs called P and A. So the initial tRNA with attached amino acid is in the P site. A new tRNA corresponding to the next codon on the mRNA binds to the A site. So ribosome catalyzes the transfer of the amino acid from P site onto the amino acid at the A site, forming a new peptide bond. So the ribosome then moves down one codon, and the now empty tRNA at the P site is displaced of the ribosome, and the tRNA that is growing peptide chain and it's moved from the A site to the P site. So this process is then repeated. The tRNA at the P site holds a peptide chain and a new tRNA binds to the A site. The peptide chain is transferred onto the amino acid attached to the A site tRNA, and the ribosome moves down one codon, displacing the empty P site tRNA and moving the tRNA with the peptide chain from the A site to the P site. This is how it works. Schematic. For termination, there are three codons called the stop codons. They code for the no amino acid, and all protein coding regions end in stop codon. So when the ribosome reaches the stop codon, there is no tRNA that binds to it. Instead, proteins called mRNA factors, release factors bind and cause the ribosome, mRNA and the new polypeptide to separate. The new polypeptide is complete. Okay. Note that the mRNA continues on past the stop codon, and the remaining portion is not translated. It is the 3' translated region or the 3' UTR. For post-translation modification, new polypeptides usually fold themselves simultaneously into their active conformation. However, some proteins are helped and guided in the folding process by chaperone proteins, and many proteins have sugars, phosphate groups, fatty acids, and other molecules covalently attached to the certain amino acids. Most of this is done in the endoplasmic reticulum. So many proteins are targeted to specific organelles within the cell. Targeting is accomplished through signal sequences on the polypeptide, and in the case of proteins that go into the endoplasmic reticulum, the signal sequence is a group of amino acids at the N-terminal of the polypeptide that is removed from the final protein after translation. Coming to the genetic code, it consists of 3 nucleotides in the mRNA. On the mRNA is a codon. Since there are 4 bases, there are 4 to the power 364 possible codons, which must code for 20 different amino acids. It is more than one codon is used for most amino acids, and the genetic code is degenerate. So this means that it is not possible to take a protein sequence and deduce exactly the base sequence of the gene it came from. So in most cases, the third base of the codon, the vowel base, can be altered without charging the amino acid. So AUG is the start codon. All proteins are initially translated with methionine in the first position, although it is often removed after translation. So there are also internal methionines in post proteins coded by the same AUG problem. So there are 3 subcodons, also called nonsense codons, and proteins end in a stop codon which code for no amino acid. This is the schematic or the table of genetic code, all the codons that code for different amino acids. So genetic code is mostly universal. It's almost universal. It's degenerate. It is used in both eukaryotes and prokaryotes. However, some variants exist mostly in the mitochondria and few genes. So CUA codes for leucine in the universal code, but in yeast mitochondria it codes for threonine. Similarly, AGA codes for arginine in the universal code, but in human and Drosophila it codes for arginine. So there are few known variants in the code in nuclei, mostly among the proteins. So the function of the different types of RNA in the cell is to facilitate the task of synthesizing proteins. An activated tRNA will with anticodon AUG bound to the serine at the acceptor stem. Okay. So the RNA in the cell facilitates the following steps. Genetic information coded by the DNA is transcribed and processed into the mature mRNA into the molecules. These molecules move out of the nucleus to the ribosomes and cytosol. mRNA leaves the nucleus and attaches to a ribosome. Where the ribosome, where genetic information in the mRNA is translated into a sequence of amino acids of a protein. Now to determine the amino acid sequence coded in the following section of the mRNA, can refer to the genetic code for the identity of these amino acids, like CCU will code for proline and GGA will code for glycine, whereas AGC and CU will code for these proteins. And mRNA section codes for the amino sequence are written in the following order. So this is the codons in the mRNA. If you can see the genetic code in the genetic code that talks about the entire system. AUG will code for the start codon. If you can find it over here, this is the start codon. It also codes for methionine, and stop codons will signal the end of the peptide chain. It is UAA, UAG, and UGA. So molecules of tRNA then pick up specific amino acids according to their anticodons, and tRNA activation occurs when the correct amino acid is attached to each tRNA. So what happens next? The start codon binds the first tRNA carrying the amino acid methionine to the mRNA. As the ribosome moves along in the mRNA, a new amino acid forms a peptide bond to the growing protein chain. So eventually, ribosome encounters a stop codon with no corresponding tRNAs. It signals the termination of polypeptide synthesis, and it's released on the ribosome. So it enters the pool of the free tRNA and ready to be recharged with a new amino acid. Okay, so this is an overview of translation. I already talked about all of this. The second stage in gene expression in translating the nucleotide sequence of messenger RNA molecule into the amino acid sequence of proteins. Okay, so tRNAs, as you know, serve as adapter molecules that couple the codons in the mRNA with the amino acids they each specify, thus aligning them in the appropriate sequence before peptide bond formation. Translation occurs on ribosomes, complexes of protein and RNA that serve as the molecular machines coordinating the interactions between mRNA, tRNA, the enzymes and the protein factors required for protein synthesis. And many proteins undergo post-translational modifications or PTM as they prepare to assume their ultimate roles in the cell. So most genetic code table designates the codes for amino acids as mRNA sequences. Important features of the genetic code are here, like they are consist of three bases like triplets. There are 64 codons. They are written in 5' to 3' direction. 61 codons code for amino acids. The other three code for the stop codons. There is one start codon AUG coding for methionine. Protein synthesis begins with methionine in eukaryotes as well, and formal methionine in prokaryotes. So code is unambiguous and each codon specifies no more than one amino acid. The code is degenerate. More than one codon can specify a single amino acid. All amino acids except MET and tryptophan have more than one codon. For those amino acids having more than one codon, the first two positions in the codon are usually the same. The base in the third position often varies. So the code is often universal. Some minor exceptions to this occur in mitochondria and some other organisms. Genetic code. Now, what is the Wobble hypothesis? So many amino acids are specified by more than one codon, right? Redundancy, which means frequently a tRNA can translate more than one of these codons, sparing the cell from making multiple tRNAs to carry the same amino acid. Now, for instance, the AG arginine tRNA can translate both CA and CGG codon that specify arginine, and this phenomenon is known as the wobble and can be summarized as below. Correct base pairing is required at the first position of the codon and the second position of the codon. Third position of the codon does not always need to be paired with the anticodon. It is allowed to wobble in that case. A mutation is a permanent, heritable, and change in the DNA base sequence of an organism. This altered DNA sequence can be reflected by changes in the base sequence of mRNA and sometimes by changes in the amino acid sequence of protein. And mutations can cause genetic diseases. They can also cause changes in amino enzyme activity, nutritional requirements, etc. So these are the common types of mutations on protein structure. If you can see over here, we have already studied all of this. Okay, I've talked about all of this. The effect of mutations is minimized in the genetic code. The mutation of the third base in the codon changes the codon meaning only 1/3 of the time. So in amino acids with only two codons, the mutation always has to be purine or pyrimidine or vice versa to change the A4 code for. This is much better, harder than purine or pyrimidine to pyrimidine solution. So the probability of getting the assignment of codons to can be calculated as here. So if we have 64 codons and we can say that each codon has an equal probability of being assigned to an amino acid, then the probability of getting any particular set of 64 assignments is this. Okay, so we'll not go into too much detail. The schematic, you can check the schematic of how translation initiation occurs. Now we have the amino acid tRNA synthetase enzyme which attaches the correct amino acid to the correct tRNA. So this is an energy consuming process, and basically what these synthetases do, they recognize tRNAs on the basis of their loop structure, not by direct recognition of the anticodon. You can see the diagram over here. So what are the requirements for translation? Ribosomes, RNA and proteins, mRNA nucleotides, tRNA, then amino acid tRNA synthetase. Protein is a product of translation and cannot be explained away by the RNA world theory. We're coming back to the question that we started with. Which of the following is the start codon? So among all of these, AUG is the start codon, which also codes for methionine, right? So if you have any questions regarding this, you can ask me now. We can move ahead to the next question in a few minutes. Moving on to the next question for today. The Shine-Dalgarno sequence is the docking site for A. RNA polymerase, B. ribosome, C. DNA polymerase, D. reverse transcriptase. Which one do you think is correct? Does anybody know what is the Shine-Dalgarno sequence? Anybody? So the Shine-Dalgano sequence is the ribosomal binding site in bacterial and archaeal messenger RNA that helps initiate protein synthesis. Okay. So the location is it's located in the ribosome binding site of mRNA, usually about 8 bases upstream of the start codon AUG, right? And the sequence aligns the ribosome with the start codon, tells the bacteria where to start protein synthesis and ensures that the genes are read correctly. Okay. So this was discovered in 1973 by Lynn Dalgarno and his PhD student John Shine. Okay. There's a lot of diversity in the Shine-Dalgarno sequences with both within and between genomes. Okay. So basically the ribosomal protein, the ribosomal protein binding site, so the ribosomal binding site in bacterial messenger RNA becomes known as the Shine-Dalgarno sequence. It basically enables initiation of protein synthesis by aligning the ribosome with the start codon. Simply put, genes are read in groups of 3 letters, but you need to let the ribosome know where to start. Okay, so the correct answer is ribosome. So, what is protein synthesis? What are the aspects? What is the mechanism? What are the differences between eukaryotes and prokaryote protein synthesis? Translational control and post-translational levels, events, etc. Okay, all of this we'll be learning. So, in the cleft of the ribosome, an anti-parallel formation of 3 base pairs occurs between the codon of the mRNA and the anticodon of the tRNA. So they explain the redundancy of the genetic code. 18 amino acids are encoded by more than one triplet codon, which usually differ at 5' anticodon base. So the 5' anticodon base is able to undergo more movement than the other two bases and can thus form non-standard base pairs as long as the distances between the ribose units are close to normal. So all possible base pairings are at the vowel position. So no purine or pyrimidine base pairs are allowed as ribose distances would be incorrect. Okay. So vowel pairing is basically non-Watson-Crick base pairing. Now what is ribosome binding site? All the Shine-Dalgarno sequence is only present in prokaryotic translation. It's a purine-rich sequence usually containing all or part of the sequence AGGU. Upstream of the initiation codon in prokaryotic mRNA, it is important to position the ribosome at correct initiation site for protein synthesis. This is a Shine-Dalgarno element. Now, each mRNA transcript is read simultaneously by more than one ribosome. So, second, third, fourth, etc. ribosomes start to read the mRNA transcript before the first ribosome has completed the synthesis of one polypeptide chain. So multiple ribosomes on a single mRNA transcript are called polyribosomes or polysomes, and multiple ribosomes cannot be positioned closer than 80 NT. So this is what we call polysomes. Electron micrographs of ribosomes actively engaged in protein synthesis revealed by beads on a string appearance. Now, methionine is the first amino acid incorporated into the protein chain in both prokaryotes, then modified to N-formyl methionine and eukaryotes. Initiated tRNAs are special tRNAs recognizing the AUG start codons in prokaryotes and eukaryotes. And initiated tRNAs differ from the one that inserts internal methionine residues. This is what the schematic looks like. So both initiated tRNA and non-initiated tRNA are charged with methionine by the same methionine tRNA synthetase to give the methionine tRNA. Only the initiated methionine tRNA is modified by transformylase to give N-formyl methionine tRNA. So protein synthesis falls into 3 stages: initiation, elongation, termination. Initiation is an assembly of a ribosome on an RNA molecule. Elongation is the repeated cycles of amino acid addition. Termination is the release of new protein chain. Now, in initiation, the prokaryotes initiation requires the large and small ribosome subunits, the mRNA, the initiated RNA, 3 initiation factors, etc. Now, IF1 initiation factor 1 and IF3 initiation factor 3 bind to a free 30th subunit. Now, IF2 complex with GTP then bind to small subunits forming a complex at RBS ribosome binding site of mRNA. Now the initiated tRNA can then bind to the complex at the P site paired with the AUG codon and release IF3. Now the 50S subunits can now bind, GTP is then hydrolyzed and IF are released to give the 70S initiation complex. So the assembled ribosome has two tRNA binding sites which are called A and P site for amino acid and peptide sites respectively. Only F-met tRNA can be used for initiation by 30S subunits. All other amino acid tRNAs are used for elongation by 70S ribosomes. So what is happens in elongation? With the formation of 70S initiation complex, the elongation cycle can begin. It involves the three factors EFTU, EFTS and EFG as well as GTP, charged tRNA and 70S initiation complex. Three steps of elongation are charged tRNA is delivered as a complex with EF2 and GTP. Now peptidal transferase, it is a 50S ribosomal subunit, makes the peptide bond by joining the two adjacent amino acid without the input of mode energy. Translocase with the energy from GTP moves the ribosome one codon along the mRNA, ejecting the uncharged tRNA and transferred the ribosome peptide from the mRNA. So peptide bond formation takes place by reaction between the polypeptide of peptidal tRNA in the P side and the amino acid of the amino acid tRNA in the A site. So then translocation in bacteria, the discharged tRNA leaves the ribosome via another site, the E site, and in eukaryotes, the discharged tRNA is expelled directly into the cytosol. Okay, so EFG translocase and GTP binds to the ribosome and the discharged tRNA is ejected from the P site in an energy consuming step. So the peptidal tRNA is moved from A site to the P site and the mRNA moves by one codon relative to the ribosome. So this is what happens. Now for termination, protein factors called release factors are there which are of prime importance, which interact with stop codon and cause release of completed polypeptide chain. For example, RF1 and RF2 recognizes the stop codon with the help of RF3. And the release factors make peptidyl transferase transfer the polypeptide to water and thus the protein is released. Release factors and EFG, they remove the uncharged tRNA and release the mRNA. Now, most of the differences in the mechanism of protein between prokaryotes and eukaryotes occur in the initiation stage, where a greater number of scanning processes involved in eukaryotes. The eukaryotic initiated tRNA does not become informylated. Okay. So the eukaryotic 40S ribosomal subunit complex binds to the 5' cap of the mRNA and moves along it, scanning for an AUG start codon. So eukaryotic ribosomes migrate from the 5' end of the mRNA to the ribosome binding site, which includes an AUG start initiation codon. In contrast to the events in prokaryotes, the initiation occurs involves the initiation tRNA binding to the 40S subunits before it can bind to the mRNA, and phosphorylation of ELF2, which delivers the initiation tRNA, is an important control point. Give me a minute, 1 second. My laptop is doing an issue. Give me a minute. हेलो हाँ आप बोलिए आप कब किधर हो अभी अभी कब तक रहोगे कैंपस में अभी हाँ 10 मिनट बाद जब H10 के पास आओगे तब कॉल कर लेना मुझे मिनिमम 10-15 मिनट लगेगा ठीक है ओके 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 sorry okay so initiation involves the initiation tRNA binding to the 40S subunits before it can bind to the mRNA. And phosphorylation of ELF2, which delivers the initiation tRNA, is an important control point. Okay. So we will not go into too much detail of this. The protein synthesis elongation cycle in prokaryotes and eukaryotes is quite similar. You can actually take a standard molecular biology book and go through it. And eukaryotes only use one release factor, which require GTP to recognize all three termination codons. And termination codon is one of the three UAG, UAA, and UGA. that causes the protein synthesis to terminate. Now, many antibiotics inhibit the protein synthesis at some specific steps. For example, streptomycin, it is a highly basic trisaccharide. It interferes with the binding of F-met-tRNA to ribosomes and thereby inhibits the initiation process. It also leads to misreading of mRNA. We have puromycin. This inhibits protein synthesis by releasing nascent polypeptide chains before the synthesis is complete. It binds to the A site on ribosome and inhibits the entry of amino acid tRNA. It acts both in bacterial and mammalian cells. We have tetracycline, which binds to the 30A subunit and inhibits binding of amino acid tRNA. This inhibits the initiation process. We have chloramphenicol, which inhibits the peptidal transferase activity of 50A subunit. It inhibits the process of elongation. We have cycloheximide, that inhibits the peptidal transferase activity of 60S ribosomal subunit in eukaryotes, and it also inhibits elongation. We have erythromycin, it binds to the 50S subunit and inhibits translocation. We have diphtheria toxin, which is given by Corynebacterium diphtheri. It binds with EF2, elongation factor 2 in eukaryotes, and blocks its capacity to carry out translocation. Okay. So there are translational control as well. Okay. So if we talk about translational control, in prokaryotes, the level of translation of different histrons can be affected by the binding of short antisense molecules and the relative stability to nucleases of parts of the polycistronic mRNA and the binding of proteins that prevent ribosome access. In eukaryotes, protein binding can also mask the mRNA and prevent translation, and repeats of the sequence 5' A U U U A 3' can make the mRNA unstable and less frequently translated. Okay. If we talk about polyprotein, it's single translation product that is cleaved to generate two or more separate proteins is called a polyprotein. Many viruses produce polyprotein. Protein targeting, the ultimate cellular location of proteins, is often determined by a specific relatively short amino acid sequence within the proteins themselves, and these sequences can be responsible for proteins being secreted, imported into the nucleus, or targeted to other organelles. Now, if you talk about eukaryotic protein targeting, targeting in eukaryotes is necessarily more complex due to the multitude of internal compartments. There are two basic forms of targeting pathways. Okay. The secretory pathway, which is the co-translational targeting. If you see over here, the signal sequence of secreted proteins causes the translating ribosome to bind factors that make the ribosome dock with the membrane and transfer the protein through the membrane as it is synthesized. So usually the signal sequence is then cleaved off by signal peptidase. Protein modification will be cleavage to remove the signal peptide, to release mature fragments from polyproteins, to remove internal peptide, as well as trimming both N and C terminal. Covalent modification can be acetylation, hydroxylation, phosphorylation, methylation, glycosylation, addition of nucleotides, all of these. Protein degradation. So different proteins can have very different half-lives. So regulatory proteins have very different half-lives. They can tend to turn over rapidly, and cells must be able to dispose of faulty and damaged proteins, right? So faulty and damaged proteins are attached to ubiquitins, with the process being called ubiquitinylation. And the ubiquitinated protein is digested by a 26S proteasome protease complex in a reaction that requires ATP and releases intact ubiquitin for reuse. Now in eukaryotes, it has been discovered that the N-terminal residue plays a critical role in the inherent stability. Okay. So what are the different methods of regulation of gene expression in eukaryotes? RNA processing. For example, gene amplification. The expression of a gene is increased several fold. This is commonly observed during the developmental stages of eukaryotic organisms. Malignant cells can develop drug resistance by increasing the number of genes for the enzyme dihydrofolate reductase. So in patients receiving methotrexate therapy, then gene rearrangement, example synthesis of light chains of immunoglobulins, gene regulation by histones and non-histone proteins, post-translational modifications of different histones. Such modified histones can regulate gene expression. Then we have class switching. In this process, one gene is switched off and a closely related gene takes up the function. Okay. So binding of regulatory proteins to DNA, helix, turn helix, all of this. Then roll up enhancers and silencers. Okay. will not. If we come back to the question that we started with, Shine Dalgarno sequence was the topic. It's basically complementary to a sequence of nucleotides at 3' and ribosomal RNA of the 38 subunit. Okay, so interaction between these complementary sequences can enhance the attachment of the 38 subunit to the AUG initiator codon. So the conclusion is whatever genetic information is carried out in the mRNA sequence is translated into the sequence of amino acids in the polypeptide gene products by intricate macromolecular structures called ribosomes. Translation process is very complex, requiring the participation of many different RNA and protein molecules. Transfer RNA molecules serve as adapters, mediating the interaction between amino acids and codons in mRNA, and process involves initiation, elongation, and termination. Okay, so coming back to the question that we started with, the Shine-Dalgarno sequence is a docking site for the ribosome. Okay, so that brings us to the end of this question and also today's session. So if you have any questions related to this, you can ask me, or we can conclude the session for today. Anybody? Okay guys, if you do not have any question related to this class, we can conclude this session. This was the last live session. Hope you had a good time in these live sessions, and I will upload all these videos on YouTube and also the slides in the Google Drive format so that you can access it. Thank you. What is there a chat? How can AUG and GUG? Okay, so the codons AUG and GUG are start codons that can code for different amino acids but can also code for the same amino acid under certain conditions. Okay, so AUG is the most common start codon and it codes for methionine in eukaryotes and a modified methionine in prokaryotes, whereas GUG normally codes for valine but can also code for methionine if it is the start codon. So in some cases, GUG can act as a start codon if AUG is absent. This means that most polypeptides begin with methionine. So genetic code is usually universal with similar codons assigned to the same amino acids in most genes. However, there are some exceptions such as when one or two of the stop codons are assigned to an amino acids. Okay. So GUG mostly codes for valine or it can also code for methionine if it is the initiator codon. Okay. I hope I'm able to answer your question. So we can conclude the session today. I'm not sure about the exam pattern. I think you have to ask the NPTEL staff for that. I suggest you talk to the NPTEL staff, probably mail them or something, and you'll get to know. Thank you so much. Thank you.
Ma'am, the details for the exam are given in the admit card.
Okay, so yeah, so as she said, the details are in the admit card. Still, if you have any queries, you can mail the NPTEL stuff. That will be a good point to start. Thank you, and all the best. Thank you. I'll, con-
Thank
I'll
you. Thank you,
conclude the
ma'am.
session. Thank you.